welcome back, heroes. Today we're going to do some bioinformatics. We're going to look at some DNA sequences and some DNA design software called APE, which is short for a plasmid editor. We're also going to do a little in silico experiment, and if you haven't heard of the term in silico before, it is short for in silicon, which computers typically have silicon for the transistors, so in computer experiments. And hopefully that in silico experiment is going to reflect something similar to what you see in the real world later on when you're working in the lab. So the first thing you have to do is download this software called APE. All right, if you search DNA plasmid editor APE in a search engine, you'll get a top hit for this software here. Click on that, it'll take you to this page. You can see a download link here for Mac, a download link here for Windows, and also a download link here for Linux. So complete the download, install the software, and we'll move on to the next step, which is looking at the DNA sequence files. All right, so before we open up the software, I want to say a few things about the DNA sequence files. All right, so there are a lot of different file types that you can get from online or create yourself that can be used to to store DNA information and to ultimately open and look at DNA information. Now, what I have here on my screen, you'll see puc19.gb and pbr322.gb. These are two different files that contain DNA genetic information, right? Now, there are a lot of different extension types. This is .gb, which stands for .genbank, okay? And GenBank is one of the largest, if not the largest, repository of DNA sequences in the world, all right? So that's very common. It contains, I think, the most information-rich file types. So you have a lot more information than some of the more basic file types. Another one you might see is called FASTA, F-A-S-T-A. It contains less information, more or less just the sequence, maybe a few other things, but nothing like GenBank, which holds all the information that the authors create about different meanings for the different sequences within that and whatnot. So let's just open one up quickly in a text editor to see what it looks like. When you double click on this file, it's showing the APE logo. So this would just open the APE software, but you don't actually see what the file looks like under the hood, okay? So we're gonna look at that first. So let's open that with another file. For example, let's open it with any sort of text editing, programming software like BBEdit or even just a Word editor. All right, so you can see it opened here. I'm going to expand, let's go up a little bit. And so you can see there's a lot of words here. It looks a little bit confusing, but maybe I'll quickly break it down for you. So at the very top, you'll see on line 1, you'll see information like the name of this, it's PUC19, how many base pairs or how long is it, what is it, this is DNA, is it linear or circular? In this case, it's a circular plasmid, so it says circular, and the date that this was created, which was this year. All right, there's a little bit of information about what this is, maybe the original author of this GenBank file, which is New England Biolabs, a company that sells restriction enzymes and other really important tools for genetic engineering. We have some let's see here, information about who submitted it to GenBank. All right. And then we have a lot more information down here about features, okay? And so we have, let's just kind of go through here. Okay, so for example, a gene. So in GenBank files, it'll have here, like, it'll say gene, and it'll say from base 146 or nucleotide 146 to 469 is a gene which is LAC-Z alpha. All right, and so that's uh, an enzyme, part of an enzyme actually, that's used for some selection. So this would tell us that in the sequence at the very end here, that shows us the full sequence of the plasmid. If we were to go from, or was it 146 to 469, that would encode for LAC-Z alpha. All right, we can see this tells us about the amino acid, so the coding sequence for the gene. Remember, a gene is more than just the information that codes for a protein, right? It has the non-coding information as well. So you can see for the gene 146 to 469, there's some coding information for protein, and this is what that is, right? These are the amino acid, the single letter amino acid designations. What else do we have here? It's got other information, a -10 signal for promoters, -35 protein binding. It has a replication origin or an ORI, which you learned about earlier. So that's from 867 to 1455, right? The PMB1 ORI. What else? So it has other things. So really, when you look at GenBank files, they give quite a bit of information about what is actually in this DNA sequence that you're going to look at, right? And because it has this information, when you open it in a program like ApE, it will also typically include this information, right? If you were to open a file like a FASTA file, which you might come across, it doesn't have all of this. And so if you were to open up that FASTA file in ApE, it would give you the sequence, it would show you that it's a plasmid, but it wouldn't be information rich like this and really show us a lot more of what's going on inside. Okay, so that's a little bit about file types for opening and plasmid editors. So now let's move on to opening this file in APE. Okay, so you'll need to find this file online. You'll probably download it from an amino folder from the amino website. And in this case, I can just double click on it. I have APE as my default program to open up a .gb file. I've double clicked it and there's that sequence. So again, we're not seeing all of that text information about what is in the sequence or what the sequence actually does and the different molecular components, but we're seeing that sequence that was at the very end, okay? I can click at many different things, but maybe we'll just do some high-level intro to this. I'm on a Mac, might be slightly different from a Windows machine, but you see a lot of the same thing. You can copy and paste things. We have things like reverse complement. So sometimes when you get a DNA sequence, you have it's in the wrong direction, right? So right now we're seeing the 5 to 3 prime in a particular direction. We might want to look at the sequence in the opposite 5 to 3 prime direction. And so you might use the reverse complement feature here. We'll be using the enzyme selector feature where we're going to do a digest shortly, because that's what this is all about. We're going to be cutting some DNA and we want to get an in silico experiment done where we can predict what we might see in a real restriction digest experiment and run it in a gel. All right. Open reading frames, you can do maps, you can designate whether you want to start with the methionine and where you want to stop. And then there are a bunch of other tools for designing primers that might come up in later videos, right? So what we're going to do today is we're going to look at restriction enzymes. And so you learned a little bit about this where these are enzymes that can very specifically bind to a DNA strand based on maybe a 4, 5, 6, 7, 8, 9, 10 nucleotide sequence, all right? And they're very specific in their binding. And once they bind to that DNA, they can actually cut that DNA in a particular way, right? So they can actually cause a cut, kind of like molecular scissors, and separate that DNA, right? And so what we can do is, let me just click on this little circle here. This is for a graphic of a plasmid, right? So this should turn up very basic, and it does. So if you were to kind of zoom in on the tube of DNA that you'll be using in the lab, there will be lots of little plasmids, all right? There'll be thousands, even millions, I would say. You could do that calculation if you wanted to. But we can see here we're working with circular DNA. It's called PUC19. It's 2,686 base pairs long. And yeah, let's try and enrich this visualization here a little bit. So if we go to enzymes and we go to enzyme selector, all right, so we have the enzyme selector open, and what you're seeing here are a lot of different restriction enzyme sites that could be in a DNA sequence, okay? And restriction enzymes, you might recall, are enzymes that are created by lots of different organisms that are actually part of their innate immunity, where if, let's say, there was a virus to try and infiltrate a cell, these restriction enzymes would be floating around with the idea that perhaps that virus that is a DNA virus, for example, it might that restriction enzyme might be able to chop up that foreign DNA and protect that cell. So there's a lot of different restriction enzymes, and they bind specifically to different target sites. And when we look here, we can see that there's a number next to a particular abbreviation, and that number gives us an idea of how many of those sites exist within the plasmid DNA that we've just loaded, okay? And so this AARL1, there's actually 0 sites in this plasmid, so if you were to try and digest this plasmid with this enzyme, it wouldn't digest at all. There's no sites, okay? We can see BAMH1, that has 1 site, etc. Okay, so that gives you some indication of what's there. Now, let's say I wanted to look at the plasmid here with an enzyme like BSA1, which is pretty common, and another very common, EcoR1 here. I would just click to select them like this, right? And then maybe I want to look at that in a map. Let's see here. Okay, so we've got our plasmid. This is what it looks like again as a plasmid, except now rather than being a blank plasmid, we actually see where these restriction sites are represented by these lines and then the name. And it actually shows us the base pair where it is located. So remember that this is the start point 0, and this plasmid is 2,686 base pairs long, which is right at the end here. And so the EcoR1 is at base pair 3, it's going to cut at 396, and then BSA1 is going to cut at 1766. Okay. And it even shows us when we hover over it, it shows us what the restriction enzyme recognition sequence is. So GGT-CTC is the sequence for BSA1, and then we've got EcoR1, which comes up as GAA-TTC. Okay, so we could imagine what this could look like in a real experiment where we cut this plasmid, this PUC19 plasmid with EcoR1 and BSA1. We could see that it would almost chop this in half, wouldn't it? So if we were to do this experiment, it might chop the plasmid in half. And even though it would create 2 different sequences from there, right? So it would cut this side, and then this side would be present as well. If we were to run that in a gel, it might actually only look like 1 band of DNA, right? That's kind of a tricky one. So maybe let's close this. Let's go back. Let's choose a different enzyme where we would have an asymmetric cut. So EcoR1, let's remove that highlighting. I'm going to choose another one called NDE1, and I'm going to open the map again. Let's make it bigger. Okay, so we can see now we're not cutting straight across to make almost exactly the same size as the DNA sequences. Now we have ND1, which is a little bit off to the side. It's at 183. And so there's a good chance if you were to cut this plasmid with BSA1 and NDE1, you would have two bands within the gel, right? So, okay, let's move on to doing an in silico experiment and see what it, with the software says it might look like. So because I've selected EcoR1 and NDE1, when I click digest right here, it's going to run such an experiment, okay? And when you do this, you need to make sure that you don't have any of the sequence here highlighted. If you do, it's going to go into linear mode and it's going to be a bit confusing. So make sure you don't have anything highlighted down here. And we're going to go click on digestion. And there we go. It's going to open up a little dialog here and give us some idea. So we have this lane one here. Do you remember what this is called? Yeah, it's called a ladder. All right, and there's many different kinds. And so in this case, if you hover over, it just tells you roughly what the in silico ladder is. So this would be a 3,000 base pair fragment of DNA, right? If we look on the left here, we have a summary of what the cut would be. So we have sizes here, for example. If we hover over here, it also highlights everything. And then if you look here, this is a linear representation of the cut. So if I hover over this top band here, the red that we're seeing on the left right here on this line, that represents the length of DNA that would be cut, right, from 183 to 1766. And if I hover over this one, this would indicate on the left again what the size of the fragment, right? And remember, it's linear, so it's showing us this linear representation, but what we're actually using in the lab is circular plasmid. Okay, so if we were to do this experiment, we should get a band that is 1583. It should be, yeah, just below the 1650 band because it's a bit smaller, so it'll run a little faster. And then we should also get an 1103 base pair band that's just above the 1000 BP ladder marker, okay? All right, so yeah, if you followed along, you've done your very first in silico DNA digest experiment. And so I urge you to use this same software and the same procedure to do an in silico experiment of the one you're going to do in your lab, okay? And so Amino Labs offers a couple of different plasmids. We didn't even look at PBR322. We don't need to. It's the same principle. You'll see it's a bigger plasmid with different restriction sites in it, but it's your choice. You get to choose between PUC19 and PBR322. And then there are also a couple of restriction enzymes that you get to choose from. So you should probably have a unique experiment compared to your peers. And so I urge you to run an in silico experiment first and then run your lab experiment. And I hope they match up.